Hello, welcome to this introduction session of our division's research program. I'm Lei Xing. I'm the Director of Medical Physics Division at Stanford Radiation Oncology Department. At Stanford, we have a very active and multidisciplinary research program in biomedical physics. As you will see from the presentations of our faculty members, our research covers a broad range of topics that are both clinically significant and novel. My lab's research is focused on AI and AI applications in biomedical physics and healthcare in general. And these are a few photos of my lab members in the last few years. What I show you here is a typical clinical workflow of radiation therapy. And we are providing AI solutions to many significant problems in this workflow. On the imaging side, we are working on data-driven image reconstruction, which promises to provide you a high-quality image with much less measurement data. In modeling, we are working on deep learning-based tumor detection, OAR segmentation, and multimodality image fusion. We also worked extensively on automated treatment planning, image-guided dose delivery, and also we're working closely with our physician colleague to develop a deep learning-based model to predict the patient treatment outcome. So this is an example of a data-driven image reconstruction. So in this work, we showed that we were able to use only one of few projection measurement to reconstruct a high-quality or volumetric image. In this year's a work done by Oscar and Shen. So they use a fine-tuned large foundation model to assess the quality of a treatment plan and also to provide feedback on which parameter should be tuned in the optimizer to really improve the quality of the treatment plan. And this work already placed in the archive, and you can find a preprint from the archive. So we are working actively with AI researchers on Stanford campus to develop a next generation multimodality and a multi-scale foundation model that is capable to take a different type of input data that include the image, pathology data, omics data, and also electronic patient record, and then to provide solutions to a number of important tasks in biomedical research and clinical practice. We do have two books on AI in medical physics and medicine. If you are interested, please check them out. Last but not least, I want to thank you again for your interest in our program, and also thank you.
My name is Ruijiang Li. I'm associate professor of radiation oncology at Stanford. I lead a group that's focused on AI for precision oncology. Lab is broadly interested in multimodal foundation AI and its applications in precision oncology. The goal is to integrate multimodal data that includes imaging, pathology, and clinical data to improve treatment decisions. So in one of the early work we did, we proposed a unifying radiologic tumor classification system that's broadly applicable across tumor histologies and imaging modalities. We've shown that tumors can be classified into multiple subtypes, and these subtypes are correlated with the distinct survival patterns and a response to immunotherapy. The other major research theme is biology-informed AI, where we leverage the existing knowledge about tumor microenvironments and develop a imaging-based model to predict this immune and stromal status, and that can increase the interpretability, and also the model will generalize better for predicting outcomes. I want to highlight one ongoing project in the lab, which is developing AI-based imaging signature for predicting head and neck outcomes. This is our unfunded project. We've collected a cohort of 400 patients at Stanford. We're planning on validating that signature in different multi-institutional cohorts. More importantly, we plan to work with NIT Oncology to test this signature on over 1,800 patients in multiple phase 3 clinical trials.
Hi, everyone. My name is Xuejun Gu. I'm the PI of Radiotherapy Innovation AI Lab, or Radiolab. Today, I'm going to give a brief introduction of our lab. So who we are. We are a group of devoted researchers with backgrounds in physics, engineering, and medicine. What is our mission? Our mission is to innovate and develop cutting-edge AI solutions to support radiotherapy clinic by enhancing professionals' capability for improved patient care. And we also support radiotherapy preclinic by facilitating research on radiobiology exploration. Our lab's first focusing is AI for clinic. We have developed AI-empowered SIS brain mets management platform. Multi-brain mets SIS require brain mets identification and distribution over sections to control treatment time and radiation toxicity. We have used AI technology to accurately and efficiently auto-segment and distribute brain mets. Currently, we are developing multi-modality AI models to predict SIS outcome for better patient care. Our second focus is AI for preclinic. Histology crypt quantification are widely accepted for studying radiation-induced damage and tissue recovery in radiobiology. We are building an AI-based histology imaging analysis platform for accurate and efficiently identify crypts and investigating crypt macrochrony for iso-effect radiation dose estimation. Finally, I would like to show a picture of our team who are from both Stanford and UT Southwestern. Our lab is well funded by NIH and with collaborators across academia and industry.
Hello and welcome. In this video, I'll be giving you an overview of the work that we do in the Physical Oncology Lab. My name is Guillaume Prats, and I'm the principal investigator of the lab. So I've been very fortunate to work with a great team of researchers, including postdoctoral fellows, graduate students. And that team is actually quite diverse. We have people who come from physics background, chemistry, biology. We work together on a number of projects. And so broadly, our lab is interested in both imaging and therapy. And I'm going to talk a little bit more in details about some of these projects. In terms of imaging, one of our projects involves trying to do PET imaging at the single cell level, which you can see on the video on the left. So this is a project that involves many different parts, such as radiochemistry, which is to label cells with a radio tracer, radiation physics to understand the way images form through radiation, image reconstruction algorithms that are necessary to achieve the level of sensitivity that is desired. And then there's a lot of research that is focused towards preclinical and clinical applications, so using the technology towards specific goals. On the therapy side, we're also interested in flash radiotherapy, and specifically, we're investigating what is the specific mechanism that gives rise to the so-called flash effect. So flash is a type of radiation given at very high speed that results in differential sparing of normal tissue compared to tumors. And in our projects, we've been looking at, for instance, how oxygen depletion and its dynamics may contribute to the flash effect. So this is through either computational models or experimental models. We're also studying how radiation chemistry may impact the response of various tissue to flash radiotherapy. And we do research in a range of different preclinical models. And finally, we have also been interested in biology-guided radiotherapy, which is an emerging type of radiotherapy that uses PET to guide the delivery of radiation. And in that area specifically, we're interested in identifying new tracers that could guide radiotherapy. We're also interested in developing algorithms to achieve real-time tracking of tumors to improve the delivery of the radiation using this type of technology. So I'd like to thank you for your attention. And please, if you have any question, you can reach me by email. Thank you.
Hi, I'm Ted Graves. I'm a faculty member in radiation oncology in the divisions of medical physics and radiation and cancer biology. And I'd like to give you a quick tour of my lab, some of the technologies we work on, and some of the science we're pursuing. So come with me. So my lab is in the Center for Clinical Sciences Research in the Stanford University School of Medicine campus. And it encompasses both wet lab and dry lab space. We have resources for in silico, in vitro, and in vivo experiments. This is one of the technologies we've engineered to perform conformal image-guided radiotherapy of small animals for basic research. The system contains an X-ray source on a rotating gantry that can both image and treat animals. We've developed methods to segment targets for irradiation, plan treatments as a series of radiation beams, and to model and verify the dose distributions received by subjects. Using this technology and others, we can investigate the efficacy of radiotherapy in treating experimental models of cancer. In this study, we compared the effects of broad field and conformal radiotherapy in terms of their ability to stimulate an immune response and ultimately to inhibit tumor growth. The effects of radiation on the immune system and how it may be combined with current immunotherapies is a specific interest of the lab. Recently, we have used conformal irradiation to demonstrate that inhibition of the protein CD47 in combination with radiation can inhibit tumor growth to tumors inside and outside the volume that was irradiated through an immune-mediated mechanism. Thanks for listening, and please feel free to contact me for more information.
Welcome to the Flash Sciences Lab at Stanford. My name is Bill Wu. I am one of the radiation oncology faculty. I'm a clinical faculty focused on thoracic radiation, particularly lung cancer. And I'm also a bioengineer laboratory scientist. I direct the Flash Sciences Lab, which is a multidisciplinary lab focused on biology, physics, and engineering, all around the concept of ultra-rapid irradiation, or FLASH. Our lab is one of the first in the country and even around the world to implement linear accelerator-based preclinical electron flash irradiation, mostly focused on mouse models. This shows our initial platform that we configured using a clinical linear accelerator. Now we've migrated this to the TrueBeam platform. This is just one example of the biological research that we have done. This in particular focused on the brain, showing the careful dosimetric characterization of our preclinical flash irradiation system, and also the biological outcomes, showing that across multiple institutions, we're able to reproduce this unique finding of flash sparing of normal brain function compared to conventional dose rate irradiation. We are looking at multiple other preclinical model systems in other organ systems. Our lab is very dynamic and multidisciplinary. We have members from students to residents and postdocs to laboratory scientists focused on different specialty areas in physics, biology, engineering, focused on simulations as well as experimental work, both developmental and in vivo biological research. I have, over the years, mentored multiple residents, both medical physics and radiation oncology residents, both as primary mentor as well as co-mentor with other faculty. Our lab also supports research in FLASH by multiple other labs across Stanford, as well as other institutions and across multiple departments. We're also developing additional platforms. This is an X-ray conformal flash irradiator for preclinical research that we are building in a new dedicated research vault that serves essentially as a linear accelerator playground and incorporates novel linear accelerator technology as well as robotic systems that we've been developing. And we're also very excited to be bringing ultra-compact proton therapy, the first of its kind, to Stanford. This will be a clinical system for patient treatment, but will also be used for preclinical flash irradiation research. Again, our collaborators span multiple departments, multiple institutions, and it's a dynamic environment that our lab members can interact with. And I hope that you'll be interested in checking us out and talking to us further. Please feel free to contact me, and I hope to talk to you soon.